Hello, I'm Harry Glorikian, and this is the Harry Glorikian Show, where we explore how technology is changing everything we know about healthcare. It feels like every week, some reporter or pundit talks about a revolution in this or that field of medicine, whether it's in diabetes management or cancer care or weight loss drugs, and obviously that word revolution gets badly overused. But a quiet revolution is exactly what's happening in the field of genetic screening of newborns. Within the last couple of years, it's become possible to sequence the entire genome of a newborn baby, all 6 billion base pairs of DNA, and diagnose potential genetic disorders in about 7 hours. That's already happening in a handful of hospitals with a focus on babies who are showing symptoms of rare genetic disorders. But within 5 years, it should be possible to extend this rapid whole genome sequencing to every baby in every hospital, whether they're showing symptoms or not. Countries like China and the United Kingdom are already investing in programs to do exactly that, though, as you'll hear in today's show, the US may have some catching up to do. My guest this week, Dr. Stephen Kingsmore, is one of the people leading this whole revolution. Dr. Kingsmore grew up and earned his medical degree in Northern Ireland, trained in internal medicine and rheumatology at Duke, studied genomic medicine at Children's Mercy Hospital in Kansas City, and now he's the president and CEO of the Institute for Genomic Medicine at Rady Children's Hospital in San Diego. At the Institute, he's been leading an aggressive push to prove that rapid whole genome sequencing and diagnosis can not only save the lives of newborns, but save the healthcare system a lot of money by making hospital stays shorter and therapies more directed. He's been able to use the argument to get Medicaid agencies in California and 5 other states, as well as a handful of private insurance companies, to cover whole genome sequencing as the new standard of care for babies who end up in intensive care with unexplained illnesses. I think genetic screening for newborns is one of the most exciting and, yes, revolutionary things happening in healthcare right now. So I was thrilled that Dr. Kingsmore could join me in early February for a detailed conversation. Dr. Kingsmore, welcome to the show.
Thank you very much, Harry. Great to be here.
So, it's interesting because I've been reading about you and the center for quite a while, and the reason I wanted to have you on the show is there's probably no one in the medical world who's more closely identified with the concept of rapid whole genome sequencing, especially in newborn infants, than you. I mean, you've published some big papers in the last few years in places like Nature, the New England Journal of Medicine, explaining how you and your team are using whole genome sequencing to diagnose critically ill newborns in less than 24 hours. And you've written that in the future, which I'm hoping is sooner rather than later, it will be possible for clinicians to order a whole genome sequencing on morning rounds and receive a molecular diagnosis by that same evening. I'm not sure how many people understand how close that future really is or how much work it took to get to this point, but our show has a pretty broad audience. So I want to maybe step back and start covering some of the real basic science here, and then we can work our way back to the healthcare implications. So if we can start with a few definitions. I mean, if you were going to explain to someone, a layperson, what is whole genome sequencing? How is it different from some of the other sequencing, techniques we've talked about? And, why do we want to do a whole genome sequence?
It's a great question. So let me start off with it. These days, post-COVID, we all know about DNA, RNA, and variants. We're not talking about a virus, we're talking about us, human beings. So packed into every cell in our bodies are 2 copies of the human genome. and it has all of the instructions for being a human being and all of the traits and attributes that you associate with me being me and you being you: blue eyes, brown hair, height, weight, intelligence. Not perfectly, but a good representation of all of that stuff is in there. And those instructions, if they were typed on regular pages like this one, it would be a book 400 feet tall. So when we say decode the whole human genome, we're talking about 6 billion letters of information, a book 400 feet tall that we are looking through, and we are looking for all of those variants, all of the differences from what we call reference, because in that information, if you have a disease, we can find out what's triggering it.
So when you say rapid whole genome sequencing, right? Rapid compared to what? I mean, how long would it have taken to sequence a whole genome, say 10 years ago or 5 years ago? And, truth be told, I was at ABI when we did the first genome. So I have a relative idea of how long it took us to do the first one. But, if you could give people orders of magnitude of time that it took us, and where are we now?
So this is like any other world speed record where the time has been coming down over the last decade. If you had asked me that 11 years ago, the answer would have been months, 6 months. If you'd asked me that 10 years ago, we set our first record, it was 48 hours. Today, it's as little as 7 hours. We can decode all of that information. And we're talking about receiving a blood sample, getting it ready to go on an instrument, so extracting the DNA, putting it on the instrument, getting the code, and then looking through all of that sequence information all the way to pinpointing a diagnosis, a disease or an illness that explains what's going on in a child. All of that is now possible in about 7 hours.
That's compared to when we did it. That's if we had said that to someone back then, they would have looked at us like we were completely gone out of our minds. But um, you know, I guess this is a good moment to acknowledge there is one company that's probably done more than many others to make large scale genome sequencing faster and more affordable, and that's Illumina, right? So I was, I know the chief medical officer there, Phil Febo. I recently had him on a talk where we were talking about their NovaSeq, and you're at Rady Children's Hospital in San Diego, and you're the head of the Institute of Genomic Medicine, and I imagine it's not a complete coincidence that Rady has an institute, given that Illumina is not far down the road. I'm assuming you guys do interact quite a bit. I don't know if you can talk about the role Illumina has played in helping you achieve some of these, speed goals.
Yeah. We have been collaborating with Illumina since 2005, believe it or not, before they acquired sequencing technology. It was an English company called Solexa.
Yep.
So our first sequencer had Solexa stamped on it, and then it became Illumina overnight. So it's been a 17-year relationship. And yes, we have walked hand in hand together across a ton of different milestones that now allow us to save the lives of critically ill children all over North America. And that continues to be a strong relationship. They're headquartered 11 miles away from our hospital. And so we have certainly weekly contact with them and contact at multiple levels. And it's been hugely synergistic. We are, at the bedside with children. They have technology. When you put those two together, you come up with solutions that really fit real world needs.
So stepping back still for the basic science question, once it looked like it was possible to sequence a person's whole genome in under 24 hours, let's say, what were the most important possibilities you saw opening up for how to use gene sequencing in the clinic?
So in that time frame, we're dealing with the most urgent situations. Let's say a baby is born and starts to seize, and that baby's seizures, those epileptic fits, are not responsive to first-line drugs. That is a medical emergency. If we can find out the cause of that child's seizure disorder within 24 hours, we will have an optimal outcome. They will not have damaged their brain substantially enough to have long-term deficits. If they continue to seize for weeks, then despite the fact that we find the cause of their condition, they are likely to have lifelong deficits. And that's
Yeah.
true of most organ systems, whether it's your heart, your kidneys, your liver, your lungs, your brain. The clock is ticking, and certain conditions, certain of the 7,300 genetic diseases, that clock needs an answer within hours. And no answer within hours means a doctor will do their best, blinded to knowing what is causing the illness, and the interventions they will give most likely are not optimal and sometimes are contraindicated. One example is the one I just gave you, a baby seizing. One of the leading causes of seizures in a newborn is something called hypoxic ischemic encephalopathy, HIE. The treatment for HIE is cool the baby's temperature down, artificially cool the baby, put the baby into almost a medical coma, and this preserves brain function, believe it or not. Now, if they have one of the genetic conditions, say a metabolic cause, that will most likely kill the baby. So you see the yin and the yang.
Right.
Right intent, I'm going to do the best for this baby. Wrong diagnosis because I didn't have a genome information, bad outcome.
Yeah, I mean, it's if we talk about rare genetic disorders, right? There's a handful of well-known genetic disorders, like I think most people have heard of cystic fibrosis or spina bifida, that I think affects, I think it's 1 in 3,000 newborns. But that's the tip of the iceberg, as you said. I mean, in a sense, right? You said, I think, over 7,000 genetic
Correct.
disorders,
Correct.
some of which are so rare, they might only affect one person in a million or even one person in a billion. Um, you know, can you help people understand, you know, the scope of these problems and why this might be an appropriate way to I like to tell people it's almost like having a Google map, which maybe oversimplifies it, but just to get people to visualize it in their own mind.
Sure. Yeah, you may be tuning into the podcast and you go, hang on, this guy's dealing with diseases that affect 1 in a million people. Why am I wasting my time listening to this? Well, the thing is, there's 7,000 of them. When you add them all together, what you find is that they affect a couple of % of the population. So they're no
Right.
longer rare, they're common. And then furthermore, when you get to pediatrics, healthcare provision for children, they're the #1 cause of death in infants. They're the #1 cause of admission to a children's hospital. If you get into the neonatal intensive care unit, at least 15% of the babies in that unit on any given day have a genetic disease. So yes, our version from our adult eyes is these are rare conditions and it's mainly babies. But we are entering a new era when we realize that we have been in our minds only looking at the piece of the iceberg that we could see above the water, and below the water was 99% of the problem. We never had a tool to find these conditions, and so we didn't know that this is what was wrong with these babies. They are much, much more common than we had suspected.
Yeah. So, I mean, just coming back around to the work you guys do every day at Rady's, right? Can you describe the work you've been doing over the past, say, 8 or 9 years to try to help rapid whole genome sequencing go mainstream? I mean, for example, there's a demonstration project in California called Project Baby Bear. And you've also been building a national network of hospitals that send you samples as part of their own rapid whole genome sequencing programs. Can you talk about that a little bit?
Sure, sure. So we first got into this a decade ago. First couple of babies, and one of them immediate impact. And we had that epiphany moment that people talk about where we realized, I'm a surfer, and I just realized this is the best surf break in the world. I'm going to hang out here. And we decoded more genomes and more genomes. And what we found is that 1 in 3 babies we sequenced, we diagnosed. And that 18% of those babies, so 1 in 5, we changed the outcome. So fully 80% of those we diagnose, we changed the treatment, and 1 in 5, we changed the outcome. Now, that may not seem earth-shattering to you, but that puts this into the realm of the top 5 ever, medical interventions. That's like penicillin for infectious disease, right? That's the sort of impact we're talking about. It's not a little thing, it's transformative. And so realizing that, what we needed to do was to industrialize this. We needed to get the message out. So first of all, we had to tell physicians about this. Doctors don't learn about this in medical school. They don't learn about it in residency yet. And so we had a provider, a healthcare provider cohort who had no idea what we were talking about, no idea what to do with a genome if you decoded it. And then we had a public who also were uncertain about genomes and DNA. And is that even safe? Are you going to clone me? All kinds of questions. And then on top of that, we had no systematized evidence base. What was our playbook, right? We were inventing a new sport and we had no playbook. So all of those things had to happen in the last decade. And now, as you say, the situation is that this is becoming standard of care. That in California and Oregon and Michigan and Minnesota and Louisiana and Maryland, Medicaid now covers this. Medicaid says this is a covered benefit if you're a Medicaid beneficiary. So does Anthem Blue Cross Blue Shield nationally. So does United Healthcare Group. So we are moving from that initial phase to early adopters. We're still not at the major part of the mainstream, but we're getting there fairly rapidly to where over the next couple of years, we can realistically expect that no matter which intensive care unit your baby winds up in, they will be ordering a genome should your baby need it. And the impact is going to be immense. The economic impact, you mentioned Baby Bear. So this was what we call an implementation study. The California politicians said, this is cool stuff. We're going to seed fund it, test a bunch of babies at 5 sites, and run the numbers, right? We want to know how many diagnoses, and we want to know how much it costs us. So we did it, and we had a 40% rate of diagnosis, so a bit better than our batting average. And we found that it saved $12,000 per baby tested. Saved, not cost, saved $12,000 per baby tested. So when we brought that back to the legislature, they went, hang on. This is an additional benefit for children. It's effective, and it saves us money. This doesn't happen, right? So we are seeing rapid uptake now, even from hawks. Budgetary hawks are recognizing this is not going to break the bank. This is a benefit that will actually decrease our costs. And the reason is really simple. Many babies and many children will stay in a hospital bed until the doctor knows what's causing the illness. A genome gives that answer in as little as a day, which can save days or even weeks off the length of a hospital stay. That's why it saves money.
Right. I mean, talk about I remember we call it the diagnostic odyssey, and it still
Yes,
exists, but
yes.
it's horrible for the parents and the child, right? But now let's talk about something called BeginNGS, and I think you pronounce it beginnings, right? Which is a research program you announced at the Institute in June of 2022, where you're screening newborns for about 400 genetic disorders. But if I understand it correctly, what's new and different about beginnings is it goes well beyond just infants who are already showing symptoms of a genetic disorder. It expands the rapid whole genome sequencing to all newborns at participating hospitals. So first off, you know, how are beginning screenings different than, I guess you would talk about, the traditional newborn screening panels?
Well, let me tell you a story. Stories are always best. So a different baby comes into our intensive care unit and is paralyzed down one side. We decode the baby's genome, and the baby indeed has one of those illnesses you mentioned, the one in a million. It's a very rare form of hemophilia. Now, there's an effective treatment. The baby needs that clotting factor to be replaced. This is pretty easy to do. It requires an infusion periodically of that factor, and that'll have to continue for the rest of the baby's life. Wonderful. Rapid diagnosis, immediate treatment. Ah, hold on. The baby's paralyzed on one side of his body. What we realized was a diagnosis in a critically ill baby was not always ideal. That what we needed to do was to get ahead of the illness. What if we could identify the illness before the baby had a stroke, before the baby had any symptoms? Could we maybe avoid them coming to hospital at all? And so we got our thinking hats on and we realized since the late 1960s, this actually had been done in the US, but for only a few conditions, about 34. The baby gets a heel prick test at birth. It goes on a filter card. It's sent to a state lab, and the baby is checked for, as I said, 30 or so genetic conditions. Some states it's more, some it's not so many. And that actually happens to 140 million babies all around planet Earth. What if we could use genomes and do that for hundreds or maybe even 1,000 disorders? Every disorder that was like the one I described, I have an effective treatment. I want to get it to the baby before they get sick, because otherwise I'm closing the door of the barn after the horse has bolted, right? So that's the concept.
So how does this work logistically? I mean, you're working with multiple institutions in multiple places. I mean, how are you making it work? What stage is the project out now? Do you know how many hospitals may be involved? I mean, a little bit more information for all the listeners would be great, because I'm sure that there's a lot of people that are, in the I may be having a child stage soon.
So the project's called Beginnings, for Begin Newborn Genome Sequencing, and it's beginning, right? So it's an apt title. We have been decoding babies' genomes rapidly in intensive care units since 2010, 2011. We're now at a point where that's being implemented all over the country. Beginnings started last year. So we have a prototype. We tested 454,000 healthy adults in England, and we tested 2,500 babies who'd already received a rapid genome because they were critically ill. And we proved out that this concept worked. It's about 80% sensitive, and it's about 99.7% specific. That's good enough to advance it. So what we're doing now is raising funds, raising awareness, forming a coalition, this Beginnings consortium. It's now about 30 organizations. I'm preparing for the very first clinical trial ever to do this. And that will start, I hope, next month.
So you talked about the cost benefit a little bit, and it was interesting because I was thinking of like, well, he's not a health economist, but I'm sure that these numbers, are up there because you're talking to politicians and insurance companies, so you got to know some of them. But what would it cost, to screen the whole population of newborns in the United States for, say, 400 different disorders?
Yeah, it's a great question. And there's a little bit of looking into the future, right? Let's understand that. And so this is not a hard-baked number. But in California, the current testing, which, as I said, so in California, I believe it's like 80 conditions total, is $210. That's the fee that every insurance plan has to pay for every baby. That's 450,000 babies in California alone a year, $210. So that's what we're aiming for. How do we get this to $210? Now, we can't get it there immediately, but you had Illumina on the show recently. I'm sure they were talking about their $100 genome, and it's not quite there yet. And it's certainly not a clinical grade genome, but that's what we're aiming for. We're skating towards a puck, and that puck is the $50 genome that really is something that's clinical grade, because that would allow us to have this amazing 15 times increase in the scope for about the same price.
And you just take that multiplication. If you said we're going to save $12,000 per child by the number of children that are diagnosed with some issue across that number, and I think it pays for itself, even above $210 if somebody were to sit down and do the math today.
Yes, that's what it will cost. I hope, but let me just say that's out there, right? Today it's expensive. Our diagnostic genomes are $8,000, so we have a ways to go, and that's why it's a very different product or service. We have to make it completely automated. We have to really be thinking about scale. So first of all, we're going from thousands of babies with the diagnostic product for neonatal intensive care units to millions of babies for beginnings. So scalability, automation, and then cost reduction are the critical elements.
Yeah, but I mean, based on everything I've, all the companies I interact with and things that I see, I just, I see it as the path is going in that direction. It's never a straight line, but we're all moving in that direction, especially if people want to do the level of screening that we keep imagining as we're going forward with other diagnostic testing, approaches that I talk to with Grail or with Illumina themselves. Let's talk about the relationship between, say, diagnosis and treatment in this world of genetic disorders. So once you get a molecular diagnosis of a rare disorder, you often have to have, either a technology to be able to treat it, or you have to have a therapeutic product to be able to treat it. Um, how is that changing with what you guys are bringing to the forefront? In other words, you talk about, I believe there's a tool you've been developing at the Institute called Genome to Treatment, or GTRX. Do you feel that as we're getting more information, there are either a device or a therapeutic that is going to meet the challenge of solving that problem that you're identifying?
Yes, so this is the other driver, the hugely exciting prospect. So there's this huge misconception amongst doctors, genetic counselors, parents, the public, that a genetic diagnosis is a fixed thing. This is not treatable, and so really all we're doing is labeling disease. And that's just plain wrong. As I said, we find 85% of the time we make a diagnosis, it's changing the management of the baby. Now, that does not mean there's a cure, but it means that there's changes in how the baby's managed. Sometimes there are cures, sometimes there are effective treatments that are not cures. There's at least 500 conditions like that. RX is doing, Genome to Treatment, is it's a website and it lists for a doctor, you just diagnosed this condition, you've never seen it before, you'll never see it again, it's very rare. Here's what a world expert would do to treat that baby. It's at your fingertips. We've got the names of the drugs, the names of the interventions, who they're indicated for, who they're contraindicated for, who should you not give this to, what's a first line treatment, what's a second line treatment, how quickly should you start it, all of that information at a single source. And what we're trying to do then is upskill frontline doctors. If frontline doctors are going to be ordering genomes, frontline doctors need to know how to manage the conditions we diagnose. And many of them are, they'll never see it more than once in a lifetime. So we got to give that to them. If we don't, we'll make a diagnosis, but the child won't receive the full benefits of the changes in management that are available for that baby that would improve their outcome.
Yeah, it's one of those. It's, whenever I talk to guests about these things, I'm like, why aren't these things just standard? I mean, if you're driving someplace and you've never gone there before, wouldn't it be great to have at least a guidepost or Google Map to help you at least get to the right place? And it's sort of, it boggles my mind that I really do wish this stuff would move a lot faster compared to as fast as it does move within the system we're in. But you're talking about N of one disorders and different approaches that you need to take to get there. Do you believe that what you're working on and the direction that you're going in will unlock, say, faster therapeutic approaches to these N of one treatments? And I call it N of one, although you said, if we're looking at, a large number of babies, it's more than an N of one because we're only seeing them, geographically as an N of one as opposed to an aggregate N of one.
Yes, so something that your listeners may or not be aware of is there's a quiet revolution happening in drug development, and it's been slowly gathering momentum over the last decade. And the epiphany that the drug companies had was we should be making drugs for these rare diseases. Traditionally, if you go back even 10 years ago, that was contrarian thinking. It was, no, we want a blockbuster drug. We want a new Viagra, a new cholesterol pill, because we want to sell 1 million pills a year, right?
Right.
Now, drug companies are realizing I can be profitable and sustainable targeting rare disease. And there's two reasons for that. Number one, the Orphan Drug Act that Congress passed gave special protections for these diseases in terms of the drugs. So the drug companies have a longer life of exclusivity. And second of all, the FDA realized we need a different rule set for judging these rare disease candidates, which has made it a lot easier. People may have heard that it takes 10 years or 12 years to develop a drug and it costs $1 or $2 billion. Well, no, the rule book has changed for these conditions, so you can do it in a couple of years and you can do it for a fraction of the cost and you have more protection. And all of that means that even though you're selling only maybe 1,000 pills a year, you still have a very valuable product. That's an immense change. Now, the issue for those companies is they need genome sequences because they need the babies to be identified to be plugged into the drug. And so there's a
Right.
bottleneck for them. They can build a treatment now using genetic therapies for many, many conditions, but how are they going to find the patients? So we now have this really interesting situation where the pharmaceutical industry needs us and we need the pharmaceutical industry, because many of the conditions don't have effective therapies. And what's hugely exciting is there are now almost 4,000 new drugs in development for rare genetic diseases. Can you imagine what it's going to look like in 5 years? And 5 years beyond that, we are going to see that we have ubiquitous genomes, timely answers, and effective therapies.
And it's got to be, I mean, if you're a physician and you're not using this, how will you ever, put the two pieces together to make that connection? I mean, you've got to employ technology to sort of practice medicine as we're going forward into this future that you're talking about.
It's a conversion experience. I think every physician I've ever talked to about this there's the before and the after. There's the before they ever got an answer on a child where there was an acute intervention, and then there's the after. And before there was some kind of healthy skepticism, and after they're believers, and they go, that was one of the more meaningful moments of my professional career. I like to do that again frequently.
You almost want to get them in one room so that we move this faster. But so, asking you just, how did you get interested in, genomic medicine, the possibilities of rapid whole genome sequencing? I mean, do you feel like your career was very clearly mapped out in advance, or was it a combination of, maybe some luck and technology advancements?
Boy, that's a deep question, Harry. Are you sure you want to go there? So I'm a person of faith, right? I'm a Christian, and I pray all the time, and I pray about research all the time, and I ask God, you created all this, help me be smart. And he talks to me, and he tells me stuff that goes, you kidding me? Are you serious? That's my secret weapon. I don't think I'm that smart. I don't think I'm that lucky. God whispers to me. I keep asking him, and he keeps giving me these nudges. And so as long as that keeps going, I'm going to keep going. The moment he shuts up, everybody's going to realize how stupid I really am.
I seriously doubt that, but no, I understand what you're talking about. I mean, every day I'm trying to keep up with what's going on and realizing what direction we're going in and the opportunities that are there, which keep pulling me in the direction. And it's funny because sometimes I wonder, why would anybody want to work in any other field? I mean, thank God they do, because we need all those people to do what they do. But I think we work in some of the most exciting and impactful space you could possibly ask. So I want to ask you to sort of make a prediction, like in what year or maybe years do you think it'll become standard of care in the average hospital, in the average US state, not meaning California, for all newborns to receive, say, broad genome sequencing panel? And on top of that, what policy decisions or government investments or research advancements could help make this happen sooner rather than later?
Well, I think the two are tied together, Harry. If indeed we have substantial support, this will happen a lot faster. So Genome England have government support, but $200–300 million, and they will have such a program, I would say, in 5 years. Something similar is happening in China. In the US, we're lagging, honestly. I'm knocking on all the doors and tapping on all the windows to seek funding support. And that really is the bottleneck. We have a technological solution. We now need to prove it out in clinical trials, and then we need to implement it. Now, for the NICU, for the neonatal intensive care unit, it took us a decade to get to where we are today. I pray it's not a decade before we realize this in the United States for all newborns. It would be possible to do it in 5 years, but you're right, there needs to be the political will to catch us up, to socialize systems where sometimes these decisions can be made much more quickly.
Yeah, you'd almost want every parent that has gone through this to make a trip to DC and visit every door and knock on every door and every window. And that might actually move it a little bit faster when they realize how many constituents there
Yes.
are that vote. So, hey, look, it's been absolutely fantastic having you guys on the show. I'm reading about what you guys are doing all the time, and these records that you guys are setting are, just it's fantastic. I hope it continues, it advances, and to be quite honest, it's an example for other whole genome sequencing approaches in many other disease states, because showing the technology works gives everybody the opportunity to start
Yep.
testing it out in other areas.
Totally.
So thank you for being here.
It's great to have been here. It's been a fun hour. You told me it was going to be fun. You were right. It was fun. Thanks a lot.
Thank you.